and welcome to our third lecture in our adaptive immunity series. And here we get to talk about some of the functions that our immunoglobulins downstream will elicit. Okay, so we've made some antibodies, we've made some IgG. What can we do with IgG? Okay, so we've made specific uh, uh, antibodies for um, said pathogen virus, okay? And that uh, Fab region will bind to a spike on the virus, okay? One of the spikes, maybe. And if enough of them bind to the spikes, they will effectively neutralize the virus. How? Well, those spikes won't be able to attach to the host anywhere using the viral spikes, right? That's why we had to learn about viral spikes. So now if we could take one of those receptors, instead of finding one of our cells and infecting us and entering, we can instead neutralize it by having a bunch of the IgGs that we have um, to neutralize it, okay? Uh, Additionally, if the uh, if the pathogen released a toxin like diphtheria, okay, releases the toxin and we can't recognize one end of it, right? We don't really recognize this end, but we do recognize this very specific region right here. The Fab binds to it, and instead of this toxin binding to a receptor that we have on our cell membrane and getting us sick, we neutralize the toxin, okay? So again, if we have a bacteria with a flagellum, okay, and we have an IgG or an IgA that knows how to target a flagellin molecule, that Fab region binds to the flagella along those flagellin molecules, effectively neutralizing the bacteria because there's going to be a lot of antibodies on it and won't be able to actually glide and rotate and perform its chemotaxis. Okay, so again, we've neutralized it. We didn't destroy it, we've just prevented it from doing what it needs to do in order to infect us. Okay, so neutralized. That is in stark comparison to opsonization. Okay, and opsonization is a word that sounds a lot like optimization, and I like to think about it like that. We're going to optimize the pathogen for destruction. Okay, so we've discovered the pathogen, we're going to opsonize it, we're going to target it, and we're going to flag it. They're like tiny little flags, right? Tiny little flags. And those flags then are going to be picked up by the Fc receptor. That's why we had to learn that was the Fc region. Okay, that Fc region is going to pick up the Fc receptor, and if enough of those are there, it's going to basically tell it to wrap around this pathogen exactly like this, okay, and engulf it, okay? And so antibodies then, in this case, serve as opsonins, and they inhibit infection by tagging the protein for destruction by the macrophages, dendritic cells, and neutrophils. Anything that can recognize those antibodies with an Fc receptor will bind to these IgGs and destroy it. Okay, so that is another way we can use our antibodies. Okay, and again, this is why we have so many IgGs in our blood and in our bodies, because of how versatile they are. So far we've seen neutralization, kind of neutralizing our things, and opsonization, which optimizes it for destruction. Two different things, seem similar but different. Okay, and then we also have agglutination. Agglutination is another way that we can facilitate destruction. Okay, if this big old mass of bacteria is all connected and linked together and floating through the blood, it's going to be picked up for destruction a lot easier. So it's going to facilitate phagocytosis in that sense, right? And we're going to be clumping them together. And note in this example, we use IgM, right? The pentameric is in 5, pentagon. 1, 2, 3, 4, 5. And that can really enhance the clumping effect that we see here. Okay, so that clumping, like I said, makes it easier to destruction. Okay, and then we can also use this agglutination process, an antibody process, when we actually do blood typing. And so blood typing becomes kind of takes advantage of this antibody relationship and this clumping. If it clumps, then it becomes it absorbs light differently, allowing us to see the reflected light in a different way than it would if it was not clumped. Okay, and again, they don't always have a positive outcome if you do a mismatched blood transfusion, and we can kind of see what blood transfusions can cause in our immune disorders lecture in the next lecture. Okay, and so in the next video we'll go over the major histocompatibility complex and antigen presenting cells.